Good day, everyone. My name is Mr. M.H. Abdullahi. This is Introduction to Biochemistry, BCH 121. The content. What is metabolism? Classification of metabolism, enzyme, common types of biological reaction, such as group transfer reaction, the formation and removal of carbon-carbon double bond, isomerization reaction, ligation reaction, hydrolysis, oxidation reduction reaction, and then finally the exercise. Now, what is metabolism? Metabolism is a set of life-sustaining chemical reactions in an organism. The word metabolism can also refer to the sum of all chemical reactions that occur in the living organism, including digestion, transport of substance in and between different cells. Now, there are three major reasons why we carry out metabolism. There are three major reasons why metabolism takes place in the human system. Now, the first reason is the conversion of food to energy to run cellular processes. Now, all the cellular processes that occurs in the system, such as your digestion, your reproduction, your growth, your excretion, all kind of cellular processes that occurs in the system occurs after carrying out a metabolism. That's the conversion of one form of substance from one form to another. Now, the second reason why we carry metabolism is the conversion of food and fuel to building blocks for protein, lipid, nucleic acid, and carbohydrates. Now, what happens? You take this food as a whole, and this food has been broken down to their building blocks so that they can be converted to macromolecules that are being used in one metabolic system or one pathways or the other in the system. Now, the third reason why we carry metabolism is to eliminate waste products. Now, you know that when you take food, you have this waste product that can be excreted out of your system. Is that also? Anytime we have this waste product, if they are not excreted in the system, once they accumulate in the system, they can lead to toxicity, causing toxicity, poison in the system. So this product or this food or whatever substance comes into your body has to be the waste product has to be excreted out in order to avoid causing toxicity in the system. So this enzyme catalyzed reaction allow organisms to grow and reproduce and maintain their structures and response to their environment. Now, metabolism can be classified into two major classes: catabolism and anabolism. Catabolism is the breakdown of compounds. For example, the breakdown of protein into amino acid. These are their building blocks. Breakdown of this whole compound into their subsequent building blocks. For protein, the building block of protein is amino acid. Then for your carbohydrates, you have can be broken down to the individual polysaccharides, disaccharides to monosaccharides. Now, the second classification of metabolism is anabolism or anabolic. That is building up, synthesis. Catabolism, breaking down, degradation. Anabolism, building up, synthesis. Building up of compound such as this macromolecule, such as your protein, your carbohydrate, and nucleic acid. As you can see from the figure below, when you take your food from your diet, these food are made up of different classes, right? Different compositions, such as they have the carbohydrate content, the lipid content, the protein content. Now, this food can be used primarily maybe to generate energy in the system so that you can carry out all your cellular activities. As you can see, the first measure is to the first from the breakdown of the food, you end up generating energy, catabolic, and then also releasing it in the form of heat, and also breaking down to the cellular individual cellular building blocks. Now, these building blocks can also undergo anabolism, whereby they form their individual macromolecules. As you can see, like building block from amino acid can end up forming protein, from your fats, your cholesterol, your triacylglyceride can end up forming your fats, from your monosaccharides, which are the building blocks of carbohydrate, can end up forming your glycogen, which is the storage form of carbohydrate in the muscles or adipose tissues. And also they can form your DNA, which are from your nucleic acid, and your RNA also from your nucleic acid. Now, the chemical reactions of metabolism are categorized into two pathways, in which one chemical is transferred through a series of steps into another chemical, each step being facilitated by a specific enzyme. Enzymes are crucial to metabolism because enzymes are catalysts. As you can remember, enzymes are catalysts that help to speed up chemical reactions without itself being used on, right? They allow a reaction to proceed more rapidly. In addition, enzymes can provide a mechanism for cells to regulate the rate of a metabolic reaction in response to changes in the cell's environment, or to signal from other cells through the activation and inhibition of the enzyme activity. What this implies is that this enzyme helps to regulate most metabolic system, most metabolic pathways or most metabolism in the system. Now, these enzymes can be used to determine if the metabolism or the metabolic pathways can proceed or can stop. Now, if you want a pathway to proceed, all you have to do is to activate this enzyme. And if you want this pathway to stop, all you have to do, sorry, is to inhibit this enzyme. So enzymes can be used to regulate metabolic reaction in response to changes in the cell. These changes can be either if the cell is lacking energy, then it wants more energy, so that then if it wants more energy, then it has to activate the enzyme. But if the cells have too much energy in the system and they want to stop the production of the energy, all they have to do is what? Inhibit the enzyme. And this, as you can see from the figure below, if an enzyme is involved in a reaction, the reaction doesn't require much energy as compared to a reaction that doesn't involve an enzyme. As you can see here, the blue line shows that this particular reaction requires an enzyme. So you see the only energy it needs is very little as compared to the red line that doesn't involve an enzyme. At the end of the day, they all end up producing the same product. That's your carbohydrates, your carbon dioxide, and water. But you see, with the one with the enzyme, only requires more energy for it to produce carbon dioxide and water, while the one without, that's the red line, the one without the enzyme requires a high energy input just to produce the same product. Now, enzyme shape is critical to the function of the enzyme as it determines the specific binding of the reactant. This occurs only by lock and key model where the reactant is the exact shape of the enzyme binding site, or by induced fit model where the contact of the reactant with the protein cause the shape of the protein to change in order to bind to the reactant. Now, looking at the figure below, what this simply means is that, like I said, the enzyme helps to facilitate the reaction, right? From A to B. Facilitated reaction, that's converting a reactant to a product. But there are two ways by which an enzyme can facilitate a reaction. The first one is using what they call a lock and key model. Now, lock and key model is just like when you have your padlock. A padlock can only open by a specific key, right? So that's what it means by lock and key. So the key has to be very specific to this to the binding site in the padlock. Now the padlock is the enzyme, while the key is the substrate. Now if the substrate enters into the padlock, it can open it because it's the key for the padlock. That's why it's called lock and key model. While induced fit model is that when you have a padlock that the key is not exactly for it, that the key doesn't specifically belongs to the padlock. But what happened is that in induced fit model is that the padlock itself will have to modify itself, induce itself so that it can conform or it can reshape itself so that it will fit the key so that the key can unlock it. As you can see in figure 1, the substrates, the purple substrate and the green enzyme, the purple substrate has exact binding site, has exact shape with the binding site in the enzyme, so it can bind equally. It can fit completely and then to activate the enzyme. But in the second figure, that's the induced fit model, when the substrate, which is green, and the enzyme, which is ash. Now the substrate binds to the enzyme, but now the substrate has to conform itself, has to to convert itself from the green to blue and yellow before it can be able to activate the enzyme to carry out the reaction. I hope it's understood. Now, within the biological system, there are 6 major classes of biological reactions that are mediated by enzymes. So enzymes can catalyze 6 different biological reactions in the human system. This includes group transfer reactions, formation/removal of carbon-carbon double bonds, isomerization reaction, ligation reaction, hydrolysis reaction, and oxidation reaction. So the first one, group transfer reaction. Now, group transfer reaction can also be called transferases. The enzymes that catalyze this reaction are also called transferases. In group transfer reaction, a functional group will be transferred from one molecule that serves as the donor molecule to another molecule that will serve as the acceptor molecule. The transfer of an amine functional group from one molecule to another is a common example, of this type of reaction. As shown here in this figure, you can see the first reactant, the acceptor is a keto acid, while the donor is an amino acid. Now the acceptor, the keto acid, has 2 carboxylic group on the carbon on the carbon 1 and 2. It has 2 carboxylic group, while the amino acid has 1 carboxylic group in the carbon 1 and then an amine group in the carbon 2. So what happens here is that the amine group from amino acid is being transferred to the keto acid, as seen in the acceptor. You see the donor now transfer the amine group from the amino acid which then converts the keto acid into an amino acid, while the amino acid that has the amine group loses its amine group, and then and then the keto acid is being transferred to the amino group, now forming a new keto acid. This is like just like they interchange themselves. This one gives the amino group, this one gives the keto group. Now the whole idea of group transfer is just to transfer a complete group. This group can either be amine, carboxylic, keto group, aldehyde group, any kind of group that can be transferred completely from one molecule to another is catalyzed by an enzyme known as transferase. A general name for the enzyme class is transferase. So they transfer a complete group from one compound to another. Now, the second kind of reaction that occurs in the biological system is the formation and removal of carbon-carbon double bond. So what happens is this: reaction that mediates the formation and removal of carbon-carbon bonds are also common in biological system and are catalyzed by a class of enzyme known as lysis. This formation or removal of carbon-carbon double bond is also used in synthetic organic chemistry reaction to create desired organic molecules. One of this type of reaction is called hydrogenation reaction. Now, one example of removal or addition of carbon-carbon double bond is hydrogenation reaction. Now, hydrogenation reaction, a hydrogen is added across a carbon-carbon double bond, reducing the carbon-carbon in double bond to a carbon-carbon single bond, as shown in this figure below. Now, let's start from the triple bonds so that we go upward so that you understand better. You see, in the triple bond now, the carbon, if you remember correctly, carbon has 4 electrons in its outermost shell. So it requires additional 4 to make it balance, right? So in the triple bond now, you see the carbon has only one hydrogen outside, and then the 3 other hydrogens are undergo what they call a covalent bond, sharing electrons so that they can have balanced electrons in the outermost shell, right? So the 1 carbon plus the 3 bonds making 4 carbons, and then their own 4 electrons in the outermost shell making 8. Now, if you add hydrogen molecule to it, the hydrogen molecule will come and displace one of the double bonds from the triple bonds to make it a double bond. Also, if you add another hydrogen to it, it can also displace the double bond from the double bond to make it a single bond. I hope you understand that. So the more hydrogen atom you add, the more it reduces the double bond. And same thing happens once you remove a hydrogen atom, it also end up removing the hydrogen in the outermost shell and then make it double bond and then triple bond. So it goes, is reversible. Now, the third kind of reaction is isomerization reaction. In isomerization reaction, the enzyme that catalyzes this isomerization reaction is also known as isomerases. There's a general name for the enzymes is isomerases. And in isomerases, a single molecule is rearranged such that it retains the same molecular formula but now has different bonding order of the atoms, forming a structural or or a stereoisomer. The conversion of glucose 6-phosphate to fructose 6-phosphate is a good example of isomerization reaction, as seen in the figure below now. Now, the glucose 6-phosphate and fructose 6-phosphate have the same number of atoms. That means they have the same number of carbon, the same number of oxygen, the same number of hydrogen. The only difference between glucose 6-phosphate and fructose 6-phosphate is the rearrangement of the keto acid there. You can see the keto acid is in carbon number 1 in glucose 6-phosphate, but in fructose 6-phosphate, the keto acid is now rearranged to carbon number 2. So by putting the keto acid carbon number 1 for in a pentose in a hexose sugar, that's hexose means 6, right? In a hexose sugar, it means that sugar is a glucose. But if you put the keto acid in carbon number 2, then the hexose sugar is now changed from a glucose to what? A fructose. But you can see that the phosphate group is still on carbon number 6. As you can see, the OPO3, that's the phosphate below in carbon number 6. They are all in the same position. It's just the keto group that is rearranged. Now, the fourth kind of reaction in biological system is ligation reaction. And the enzyme that catalyzes reaction are known as ligases. Ligases. Now, ligation reaction use the energy of ATP to join 2 molecules together. Ligase is a class of enzyme that brings about binding or joining of 2 molecules. It is a class of enzyme that catalyzes the binding or joining of 2 macromolecules by forming a new bond like carbon oxygen, carbon nitrogen, and carbon sulfur while in the cells. While in the cells, ligase repairs single and double strand break that occur during DNA replication. Ligase enzyme catalyze ligase reaction. For example, the example below depicts the ligation of 2 sticky ends that were generated by E. coli. This is a DNA gene, that's the DNA component of E. coli, Escherichia coli, if you is a microorganism. So ligase is just like ligase is the opposite of your remover and additional remover of the carbon double bond. That's the lysis. The lysis splits, the ligase join. As you can see, the two different strands are being joined by an enzyme known as a ligase, form a single double-stranded DNA molecule. I hope that one is clear. Then the fifth one is hydrolysis reaction. Now, the classification of hydrolysis reaction include both the forward reaction that involve the addition of water to a molecule to break it apart, or the reverse reaction involve the removal of water to join molecules together. Same dehydration synthesis or condensation. When water is added to a molecule to break it apart into two molecules, this reaction is called hydrolysis. The term lysis means to break apart, and the term hydro means water. Thus, hydrolysis means to break apart with water. The reverse of that reaction involves the removal of water from two molecules to join them together into a large molecule. Since the two molecules are losing water, they are dehydrated. Thus, the formation of molecules through the removal of water is known as dehydration synthesis. Since water is also a byproduct of this reaction, they are also commonly referred to as condensation reaction. The formation of the major classes of macromolecules in the body, such as protein, carbohydrate, lipid, and nucleic acid, are formed through dehydration synthesis, where water is removed from the molecules. During normal digestion, our food molecules, the major macromolecules, are broken down into their building blocks through the process of hydrolysis. Now, let me explain this in a more simple term. It means that as long as a reaction involves either the removal or addition of water, the reaction is known as a what? A hydrolysis reaction. If you are trying to split a particular compound into two, and at the end of the day you end up releasing water, that particular reaction is known as what? Hydrolysis reaction. But if you are trying to join two compound into two, at the end of the day you end up adding in water, then that reaction is known as what? That no, okay, sorry. When you're trying to split a compound into two, and at the end of the day you need to what? You need to add in water. That reaction is known as hydrolysis. But once you are splitting a single compound into two, and at the end of the day you end up what? Removing water. That reaction is known as what? Dehydration synthesis or what? Condensation. Okay, like for example, look at this in this figure here. Let's assume this is any of our macromolecule, either it being protein, carbohydrate, or lipids. Now, once you are trying to break these whole macromolecules into their individual single block, you have to add in water to it. So adding in water, that reaction becomes what? Hydrolysis. But once you are trying to join in these building blocks to form a whole molecule, that's either your RNA, your DNA, your fats, your carbohydrates, your glycogen. Now that at the end of the day, you end up having to remove the water you added when you're breaking it down, and that reaction becomes what? Dehydration synthesis. Now, remember, there's a difference between your lysis, your ligases, and your hydrolysis and dehydration. Your lysis and ligases doesn't involve water in any way. But once a reaction involves water, then it becomes hydrolysis or what? Dehydration synthesis. Now, for example, like these are examples of dehydration synthesis I have here. Like in your carboxylic acid, the first one, your ester bonds, carboxylic acid plus alcohol. You can see the R group of the alcohol binds to the carboxylic acid, binds to the OH site of the carboxylic acid at the same time releasing what? Water. You can see the only thing that goes out here is the hydrogen group. The hydrogen group in the carboxylic acid now goes and binds to the OH of the alcohol, releasing water, while the R group now binds to the carboxylic acid. The same thing happens for the phosphoester, for the phosphoester, phosphodiester, phosphotriester, and the amine group. You can see in the amine group also the carboxylic acid binds to an amine. What happens is that the amine groups comes and bind to the OH site of the carboxylic acid, while the hydrogen there binds to binds to the OH side of the carboxylic acid, while the O there binds to the hydrogen of the amino group to release what? Water. Now the same thing also happens for the carbohydrates. Now the last class of reaction we have in the biological system is the oxidation reduction reaction. Now oxidation reduction reaction is also known as redox reaction, while the enzyme that catalyzes this reaction has a general name of oxidoreductases. Oxidoreductases. The general name of enzymes that catalyze oxidation reduction reaction are known as what? Oxidoreduction reductases. While instead of to say oxidation reduction, It's a kind of very long. You can just say redox reaction. Now, redox reaction is a type of chemical reaction that involves the transfer of electrons between two atoms or compound. The substance that loses and the electron is said to be oxidized, while the substance that gains electron is said to be reduced. Redox reaction always have to occur together. If one molecule is oxidized, the other molecule has to be reduced. Electrons don't appear out of thin air to be added to a compound. They always have to come from somewhere. Now, what it means is that oxidation and reduction, they are two different processes. They are two different reactions, but they always occur simultaneously because it's just like a transfer of something from one place to another. So that stuff has to come from somewhere before it can go to the other place. So it means that when you're working with two compounds and then you have to transfer electrons, then the electron has to come from one compound. Now the compound that loses electron is considered to be what? Oxidized, while the one that gains electron is considered to be what? Reduced. And the oxidation in electrons composition can be evaluated in the change of the oxidation states or oxidation number of an atom. Therefore, the oxidation reduction reaction is any chemical reaction in which the oxidation state or oxidation number of a molecule, atom, or ion changes by gaining or losing an electron. Overall, redox reactions are common and vital to some of the basic functions of life, such as photosynthesis, respiration, combustion, corrosion, and then rusting. Now, with this figure, I use this figure to simplify, to make it more self-explanatory. Now, as shown in the figure below, an easy mnemonic for helping you remember which member gains electron and which member loses electron is Leo, the lion says gear. So you can always put that in your mind. Leo, the lion says gear. Leo stands for lose electron. And a compound loses electron, it's automatically become what? Oxidized. Gear stands for gains electron. Now, once a compound gains electron, the compound becomes what? Reduced. As stated here, Leo, lose electron is equal to oxidized. The lion says gear, gains electron, it's equal to what? Reduce. Now, there are certain rules that have been assigned for oxidation state of a compound or a molecule of an ion. Now, the oxidation state of any element corresponds to the number of the electrons that the atom loses or gains during an ionic bond. Ionic bond means direct transfer or appears to lose or gain when joining in covalent bond. Covalent bond means what? Sharing of atoms with other atoms in the compound. In determining the oxidation state of an atom, there are 7 guidelines to follow. Now, the first guideline is the oxidation state of an atom in its elemental form is 0. So all atoms in their elemental form are what? The oxidation state is what? 0. Oxidation state can only be stated in corresponding to the atoms they gain or what? Loses. This includes elemental forms that occur as diatomic molecules. For example, we know that our oxygen doesn't occur as a single molecule. It occurs as what? O2. Even our nitrogen occurs at what? N2, atmospheric nitrogen, and then your oxygen. Now, the second rule is this. The oxidation state of all atoms in neutral species is 0. In an ion, while in an ion is equal to the ion charge. For example, the overall oxidation state of a sodium chloride is 0, even though the oxidation state of sodium in this bond is +1 and the oxidation state of chlorine is -1. So +1 -1 is what? 0. 1 cancel 1, right? Now, when you add the two together, they are equal to 0. Now, in the case of ion, the overall charge is always indicated. They are always indicated above. For example, the overall charge of hydroxide is -1, while the oxygen in that hydroxide has -2 oxidation number and the hydrogen has +1 oxidation number. I don't know if you understand that. You know, oxygen has 6 electrons in the atmosphere, so it requires 2. So the 2 is requires addition gives it that -1. While hydrogen only has only 1 electron in the atmosphere, so it has to give out that 1. Giving out that 1 becomes what? +1. So when you add the compound together, hydrogen chloride, -2 + 1 gives you what? An atomic number of what? -1 for an ion. Now, group 3. Group 1 metals, which are alkaline metals, right, have an oxidation number of +1 because we know that all group 1 metals have 1 electron in the atmosphere, and group 2 metals have oxidation number of +2 when they are involved in ionic bonding because all of them have what? Only they have 2 electrons in the atmosphere, and that's how it goes. Now, the fourth rule is that the oxidation state for fluorine is what? -1. Why? Because fluorine has 7 electrons in the atmosphere and requires what? 1. So once you put +1 for them, it gains electron and becomes what? Reduced, right? Once it gains electron, it becomes what? Reduced. That's -1. Now, hydrogen generally has an oxidation number of +1 because it has only one electron in the atmosphere, right? It becomes oxidized. Then oxygen generally has -2 because it has 6 electrons in the atmosphere and requires 2. So adding 2 to it becomes what? Reduce. In binary metals compound, group 17 or 7A element have an oxidation number of -1. Group 16 or 6A have oxidation number of -2. Group 15 or 5A have oxidation number of -3. Yeah, while the seventh row is the oxidation state of other atoms are calculated based on the 1 to 7 row, right? Either if they have a 1 in the atmosphere or 1 to 7. So if they have 1, they require 7. They have 2, they require 6. If they have 3, they require what? 5. If they have 4, they require 4. If they have 5, they require 3, right? If they have 6, they require 2. If they have 7, they require 1. 8, balanced electron structure. So 8 for 8 does not require anything. Now, these are some examples I gave you how to calculate oxidation states. Now, for example, what is the oxidation state of chromium in the chromate anion? Chromium is what? Tr, chromium oxide, right? The solution, looking at the oxidation state rules, we see that the overall oxidation state is equal to the charge of the ion. Now, that oxygen usually takes -2 oxidation states. With this information, we can set up an oxidation state equation to calculate the oxidation state of chromium. Now, the oxidation state of chromium plus the oxidation state of 4 oxygen equals to -2. Now, chromium plus 4, because we know that there are 4 oxygen in that compound, right? So 4 minus 2, that's equals to -2, because they said, what did they say? They said looking at oxidation state, we see that the overall oxidation state is equal to the charge of the ion. So whenever you have its ion, the overall oxidation state is equal to the charge. So now we're just trying to see to determine the oxidation state of chromium in this particular compound. We already know the oxidation state of what? Of oxygen, right? That one is common. We know that it's -2 because it has 6 electrons in the atmosphere, so it requires 2. And then we know that there are 4 oxygen, and then the overall compound has oxidation state of -2. So we can use that to calculate for chromium. So all we have to do is chromium will be our x, right? Then 4 oxygen minus 2, which is the oxidation state of the oxygen, equals to -2, which is the overall oxidation state of the compound. So chromium minus 8 equals to -2, which is the oxidation state. Then chromium equals to -2 plus 8, so chromium becomes +6. I hope you understand. So these are other exercises I gave you below. So please, you can go and practice it. And then anybody that has any question can contact me, or can come to my office. Please. Thank you. Have a good day.